Good morning. First of all, I would like to thank Natalie for the very generous invitation to this symposium. I'm really excited to participate in it. So without further ado, I will move on to share my screen. Okay. So two major events occurred during the history of life in our planet. One was the conquering of dry land from the oceans. And the second one, I would say, was the growth and expansion of our forebrains. Now, the cerebral cortex is the part of our forebrain that is the largest and is central for our higher cognitive functions. During evolution, there was a dramatic expansion in the size of the cerebral cortex, particularly in amniotes. And this expansion is the result of developmental processes occurring in the embryo and fetus, and are mostly related to changes in the degree of neurogenesis of the cerebral cortex, with a very dramatic increase, particularly in humans. Now, the embryonic development of the cerebral cortex starts with the progenitor cells that we call radial glia cells. These are the primary progenitor cells, and they are localized in the ventricular zone. They have their cell body there, they express PAC6, and they extend these very thin processes to the surface of the cortex. Now, radial glia cells are the major source of neurons in the cerebral cortex, and they produce these neurons in two different ways. One that we call direct neurogenesis, in which upon cell division, these neurons will simply migrate away to their final position where they differentiate, or indirectly via producing intermediate progenitor cells that will localize and divide in a different layer, what we call the subventricular zone, in a basal position. And from that division, two neurons will be born that then will resume their migration and differentiate. So from a theoretical point of view, if we had two progenitors in two species, one undergoing only direct neurogenesis, given an amount of developmental time and a number of cell divisions, the number of neurons produced will be relatively small. But if neurogenesis is indirect, with the initial phase of progenitor expansion, given the same amount of developmental time and the same number of cell divisions, the number of neurons produced is much larger. Now, if we even increase further this developmental time, then obviously the amount of neurons produced and the size of the resulting cerebral cortex can be much, much larger. But before neurogenesis occurs, the radial glia cells, the neural stem cells, must first amplify so that in the end, the appropriate numbers of neurons can be produced. And this is a very important step. And indeed, the degree of amplification of this early primordium of the cerebral cortex is very different also between different species. And obviously, it has a very important consequence on the final size of the cerebral cortex. Now, following this expansion of the early cortical primordium, which is composed mainly by neuroepithelial progenitor cells, then radial glia cells are born, and from them those intermediate progenitors I was mentioning, and the postmitotic neurons. Now, this is a situation in the mouse, which is a species with a relatively small brain. What happens in other species? Well, in humans, for example, they have a very similar structure, but the subventricular zone is not as small as in the mouse, having not so many intermediate progenitors, but actually is much larger, much thicker, and it's actually subdivided into an inner portion, the inner subventricular zone, and an outer portion, the outer subventricular zone. These two subventricular zones contain a very large amount of intermediate progenitor cells like mouse, but also contain other types of progenitor cells, including basal radial glia cells that are very abundant and important, and also other types like, for example, subapical progenitor cells. So it will be this increased divergence, diversity, and high abundance of progenitor cells that will make the difference between the small and smooth cortex in the mouse and the large and folded cortex in humans. Now, usually, the evolutionary expansion of the cerebral cortex that I started mentioning is viewed as a process where some kind of a stem mammal resembling a mouse with a small cortex just increased neurogenesis and produced a bigger brain, like for example in a chimpanzee, and that continued evolving to human. However, the process is not quite exactly like this. Let me explain. When we look across mammalian phylogeny, we look at all major clades, and we look at the appearance of the brains in those species, we see that large unfolded brains are present across all the phylogeny in mammals, including primates, as I mentioned, ungulates, carnivores, elephants, even rodents. For example, the capybara has a very large brain. And we can find large unfolded brains also in marsupials, like the gray kangaroo or the wombat. And even in a monotreme, the echidna has a large unfolded brain. This means that probably the stem mammal that gave rise to all the extant mammals that we find today in our planet probably also had a relatively large cortex with maybe some degree of folding. And so later, during mammalian speciation, these mechanisms of brain development that give rise to a very large unfolded cortex were just carried over and exploited by different species in different ways. Now, when we compare the size, for example, of primates, we see that not all of them have a very large cortex like humans or chimpanzees. But actually, there's some primates, like, for example, New World monkeys, that have relatively small brains and even with an almost smooth cortex, something that we would find in a mouse, for example. So we have been intrigued by this question, and many years ago, teamed up with Wilhelm Huttner in Germany, where we compared the degree of folding of the cortex in these different primates, old world primates and new world primates. And we also took into account the phylogenic relationships between all these species. And what resulted was this. This is a representation of the phylogenic tree of these primates, indicating with color codes the degree of folding of their cortex. And what we find is that there was an ancestral primate that have a certain degree of cortex folding, and from this ancestral primate, there were two trees of evolution. On one tree, there was an increase in the degree of folding and the degree of size of this cortex, leading to primates and to humans. But there was another branch where cortex size and folding did not increase, but actually was reduced. And this is what we call a secondary reduction in brain size and folding. So this is something that we can measure and we can observe in primates. But it occurs not only in primates, but actually in all mammals. And there's where we find that in small rodents, there was a secondary loss of brain size and brain folding, and that's how we find today the small rodents with a small and smooth cortex. Now, how is the size of these brains regulated? Because obviously, it's not good just to have a big brain. Probably if mice decided to evolve or evolution drove mice to have small brains, is because that's important to them. So it's important to have the correct brain size. How is this brain size then regulated? So obviously we turn to genomics and we turn to evolution of the genomes, and actually studies again from the lab of Wilhelm Huttner have identified, and also a number of other labs, including Pierre van der Hagen's lab in Belgium, they've identified some genes that are human specific, that only exist in humans because they evolved very recently in human history. And here you have a list of genes that have been identified that are human specific and highly expressed in the developing human cortex. And for example, there's been studies again by Wilhelm and by Pierre focus on, for example, these two genes that have very important functions in the expansion and folding of the cortex, possibly important for the recent evolution of the human brain in apes and primates. Now, evolution and expansion of the cortex involve not only the occurrence of new genes, but also in the regulation of expression of conserved genes that were already existing in previous species. And this is something that we identified a couple of years ago. That's the case, for example, for the expression of ROBO and Delta-1. These are two genes that are highly conserved across phylogeny, but the expression level of these genes in the developing embryonic cortex is very different between species. For example, in reptiles, we find that these radial glia cells express high levels of ROBO and low levels of delta. And the result of this combination is that these radial glia cells will undergo only direct neurogenesis, so that the resulting cortex is relatively small. Now, in comparison, in mammals, for example, in mice, we find that in the embryonic cortex, there is low levels of overexpression and high levels of delta 1. And this makes these radial glia cells to undergo indirect neurogenesis, producing then a much larger number of cortical neurons. And this is what we find again in mammals, including humans. So regulation of the expression levels of conserved genes is also an important mechanism. Now, the genome that is related to coding genes, that is associated to coding genes, 95 to 98% of the DNA in these regions is not coding DNA, but is actually regulatory DNA that is involved in regulating the expression levels of the genes that it is associated. Now, in addition to enhancers, promoters, and distal regulatory elements, there is also additional mechanisms to control the expression levels of coding genes, including, for example, microRNAs. MicroRNAs, as you all know, are very important because they bind primarily to the 3' UTR of coding genes, and by this binding in a very sequence-specific manner, they inhibit translation of this gene or directly produce decay of this RNA degradation. So in the end, blocking and impairing expression of these genes. And actually, recently, we have identified LED7 microRNAs, for example, as being very important in the really early development of the cortex in those neuroepithelial cells where they have to expand. So microRNAs are important in early brain development. So to understand mechanisms that will be involved in that secondary reduction of cortex size that occurred in some species, like, for example, rodents, we decided to focus on microRNAs. And this has been the work of the PhD thesis of Kavya Chinappa, a student from India that we've had in our lab. She's not with us anymore. And so with Kavya, we decided to focus on this particular microRNA, MIR3607. The reason being because of the initial observations in this microRNA. We observed by in situ hybridization that this microRNA is very highly expressed in the embryonic human cortex, in the germinal layers, all three of them, ventricular, inner subventricular, and outer subventricular zone, as you can see here. By looking into published data, this is data from Collet and Lyon, published a few years ago, we found that the same microRNA is also highly expressed in the germinal layers of the embryonic macaque cortex. And also in our hands, we identified that the same microRNA is also expressed in the germinal zones of the developing ferret cortex. At early embryonic stages, we find it expressed in the ventricular zone. Later on, we find it highly expressed in the outer subventricular zone. Now, as you can see, these three species are relatively remotely related in phylogeny, but the mouse had derived from this other branch in phylogeny much more recently than, for example, ferret. However, we observed that microRNA 3607 is not expressed in the embryonic mouse cortex. So that necessarily means that there was, in this branch of mammalian evolution, there was a secondary loss of expression of this particular microRNA in cortex development. Now, so we took advantage of this circumstance to study what may be the function in cortex development of this microRNA by expressing it in the developing mouse cortex. So we have used plasmids expressing encoding for this microRNA, and you can see here when we express it by in utero electroporation in mouse embryos, we can then readily detect this microRNA. So we can study the function of expressing this microRNA. Now, importantly, the sequence of this microRNA is very highly conserved across mammals, and particularly the seed sequence, which is the most important part that confers the highest specificity of this microRNA. You can see the similarity with the human sequence that is nearly 100% for all the primates, obviously, and is also very high for hominids and for carnivores, where, for example, for mouse and ferret, the homology of the consensus seed sequence is 100%. And actually, not only in these clades, but also in ungulates and pitibagi in all mammals, the sequence of this microRNA, which actually is normal, is highly conserved. Now, what Kavya first did was then to overexpress this microRNA in the embryonic mouse cortex as this intermediate developmental stage. And what she found was initially, it seemed not so much of a change, but somewhat an accumulation of progenitor cells in the ventricular zone of these electroporated mice. However, when she looked at the behavior of these progenitors, she found that there was a higher incidence of progenitors incorporating BRDU, meaning that cycling progenitors in this ventricular zone. And then when she actually measured the rate of cell cycle exit by combining these three markers, she found that progenitors expressing this microRNA were exiting in a very low proportion the cell cycle, meaning that they were re-entering the cell cycle, not producing neurons, but amplifying themselves. Now, when Kavya looked for the expression of these two transcription factors, PAX6 and TBR2, which label, as I introduced in the beginning, label radial glia cells for PAX6 and intermediate progenitor cells for TBR2, she found that there was a very dramatic increase in the proportion of our electroporated cells that would express PAX6, particularly, and also an increase in those expressing TBR2. Now, this is very important because, as I mentioned, radial glia cells are mostly located in the ventricular zone where they express PAX6, but they're not really located in the subventricular zone or intermediate zone. And there we were seeing high levels of expression of PAX6. And also, looking at the proportions, we could see that it seemed that at least a very significant amount of progenitor cells might be co-expressing both PAX6 and TBR2. So Kavya looked at the expression levels at the protein level of individual cells, and she plotted that here, comparing the expression levels of PAX6 and TBR2. And she found that the abundance of cells co-expressing these two transcription factors, which is quite rare in mice, was significantly increased when we co-expressed, when we overexpressed this miR3607. And this co-expression of PAX6 and TBR2 is a feature typical of basal radial glia cells, that other type of radial glia cells that we find particularly in the OSVZ of primates and also in ferret. Now, the reason why we had this high co-expression of PAX6 and TBR2 is because the expression of miR3607 increased very significantly the intensity, the levels of expression of PAX6. And so now cells would co-express the two transcription factors. Now, importantly also, it's already been published and studied that overexpression of PAX6 in mouse leads to the increased proliferation of progenitors. So this result came together with the previous result I showed, that the expression of this microRNA promotes not only PAX6 expression, but also amplification of these progenitors. Now, to know what's the transcriptomic effect of this miR that would cause these changes in progenitors, we performed RNA-seq analysis following the standard protocol, following electroporation of our plasmids, either control or the microRNA. We FAC sorted the green cells and then performed RNA-seq. We found a number of differentially expressed genes, and as expected, the majority of these differentially expressed genes were downregulated. I say as expected because of the known action of microRNAs, reducing transcription or expression of genes. When we looked at the ontology of these genes differentially expressed, we found features related to progenitor cells, and specifically we found effects on Wnt signaling, on the canonical Wnt signaling, regulation of neuroprecursor proliferation, and particularly on cell fate commitment. When we looked at gene set enrichment analysis, we found, again consistent with the previous analysis, increase in terms related to cell division, on Wnt beta-catenin signaling, and interestingly, a loss in apical junction pathways. Now, when we looked at functional networks, again, we find enrichment in terms related to neuroblast proliferation and neuroprecursor cell proliferation. So it seemed that this microRNA was indeed changing progenitor cells in their behavior. So we decided to focus on earlier stages of development. So we electroporated mouse embryos at an earlier stage, E12, and then we analyzed what happens just 1 day later. And what we found is that the very nice laminar organization of this cortical primordium with cycling progenitors forming these beautiful layers was completely disorganized in the overexpression of this microRNA, forming some kind of circular structures where we could not see anymore the proper localization of these cells. Now, when we looked at the expression of TBR2, a marker of intermediate progenitor cells that should be forming this subventricular zone here, again, we found that this was very much disorganized. And when we looked at part 3, which labels the apical surface of this cortical primordium, it should be labeling the apical part of radial glia cells. This was again completely disorganized. It was not present here, but it was found within the cortical parenchyma. Now, when we looked at pH3, which labels mitosis, again, in a normal situation, we would find this apical mitosis of radial glia cells here, and this basal mitosis of intermediate progenitor forming this distinct layer. but this is absolutely disorganized in the expression of this microRNA. And this was consistent then maybe with these apical radial glia cells being delaminated into the cortical tissue. Now, indeed, when we looked in detail at the morphology of these electroporated cells, when we expressed microRNA 3607, we found that this apical side of the cortex, this part 3 staining was localized within the parenchyma, forming actually these little circles around which we would find the radial glia cells still with their normal morphology, but now acquiring kind of a cartwheel organization. And this is very different from a control situation where we can see, as again, the normal morphology of radial glia cells, but they would stand parallel to each other, not forming these cartwheel configurations. So the overexpression of this microRNA was causing the amplification of radial glia cells, but not their loss of polarity or disorganization, or actually just simply their delamination from the apical side, but they maintain their correct laminar polar structure. Now we looked for longer-term effects of expressing this microRNA from early stages, and we found just very similar phenotype, but even more exaggerated, with a more massive basal delamination of these apical radial glia cells to more inside of the cortical parenchyma. You can see here the part 3, the apical side of these radial glia cells. Again, the PAC6 stain was even further into the tissue, as was also TBR2. When we looked at phosphohistone 3 for mitosis, again, as you can see here, these were heavily delaminated into the parenchymal tissue. And actually, when we quantified the abundance of these apical radial glia cells, not surprisingly, we saw a very dramatic reduction in the density of apical radial glia cells and a very dramatic increase in basal mitosis, with a total significant increase in the abundance of mitotic activity. Now, importantly, this basal increase in basal mitosis does not really correspond to intermediate progenitor cells, but it corresponds mostly to these delaminated apical radial glia cells. And when we look at the morphology of these cells, again, we see this cartwheel configuration with radial glia cells keeping their normal polarity, but acquiring this new disposition. Now, in our transcriptomic analysis, again, we found an enrichment in the Wnt signaling pathway, as you can see here, and that involved a number of key genes in this pathway, particularly Wnt beta-catenin B1, as you can see here, that was highly expressed. And also we could see by the individual or specific tracks of expression, we could see a very significant increase in the levels of expression of beta-catenin 1. And indeed, not only we found enrichment in terms related to Wnt beta-catenin, but also in pluripotency of stem cells. Again, consistent with our previous results. No? But this led us to focus maybe on beta-catenin 1, thinking that maybe the primary effect of expressing miR3607 was to upregulate Wnt beta-catenin 1. And in fact, when we looked at the levels of activated beta-catenin in our electroporated cortex, we would find a very significant increase in the intensity of signal of this activated beta-catenin in the cortical tissue. So what we did was to, on purpose, overexpress and constitutively active beta-catenin form. And what we found is that the cortex, again, was severely altered in these electroporated embryos with the disorganization of these radial glia cells, these ventricular zone, as you can see here, forming, again, these cartwheel configurations, these kind of rosettes of apical radial glia cells, very much similar to the phenotype we observed upon overexpression of miR3607. Now, beta-catenin pathway is regulated by a protein complex that phosphorylates beta-catenin and drives it for degradation. Now, upon Wnt binding to the freezer receptors, this sequesters this protein complex to the cellular membrane so that it does not phosphorylate beta-catenin, and now beta-catenin is not degraded, so it can enter the nucleus and have its transcriptional effects. Now, interestingly, among our differential expressions after overexpressing our microRNA, we found a number of genes that are predicted targets for this microRNA, and this included APC, which, as you can see here, is one of the known key effectors of this protein complex driving beta-catenin degradation. So if this miR3607 is blocking, downregulating the expression of APC, maybe that's the mechanism by which it liberates beta-catenin from degradation, and then it increases beta-catenin signaling. And in fact, when we looked at our individual tracks from the RNA-seq analysis, we found that the levels of transcription of APC are also significantly reduced when we overexpress this microRNA. So then what we did was to look at the presence of these target sequences for our microRNA on the 3' UTR of APC, and we found that there is 2 prediction sites in the human APC sequence and its 3' UTR, and actually we found that for this octamer, this is highly well conserved across mammals, as you can see here in rodents, ungulates, carnivores, and even in bats, is very, very significantly conserved, maybe only with 1 single nucleotide change in carnivores and rodents, meaning that this microRNA can really faithfully target APC across phylogeny. And in fact, when we express this microRNA in cell culture, we could see that this was sufficient to downregulate, to reduce the levels of APC protein in culture, meaning that indeed this microRNA is not only predicted to target, but it targets APC, and it prevents or limits the expression levels of APC protein. So what we did was to go back to our experiments where expression of microRNA drives this massive disorganization and amplification of apical radial glia in ectopic positions, and now we co-expressed APC to see if that would maybe rescue the effects of the microRNA. And in fact, that's exactly what we found, that co-expressing both will completely rescue this mislocalization of apical radial glia cells incorporating BRDU, and they're back localized to their normal location here in apical positions, as we find in a control situation where expression of this excess of APC has absolutely no effect. We find this to correct not only the position of BRDU incorporating cells, but also the position of radial glia cells expressing PAC6, which are not localized basally anymore. And also we correct the apical disruption of these apical junctions that are not going into the parenchyma anymore. This is perfectly corrected. So it is the decreased expression of APC caused by the overexpression of microRNA 607 that causes this very severe phenotype. Now, as I mentioned in the beginning, this microRNA is naturally not expressed in rodents, but it is expressed in these other species. Now we have seen what happens when we express this microRNA in a mouse, but we wanted obviously to understand what is the endogenous function of this miR3607. So we turned to perform experiments in our other preferred animal model in the lab, ferrets, where since it is expressed in the developing ferret cortex, there we generated loss of function experiments. Now for these loss of function experiments, we generated tough decoys against miR3607, which are some kind of sponges that will sequester the microRNA. And as you can see here, the endogenous levels of expression of miR3607 in ferret cells are dramatically reduced when we overexpress this tough decoy, but not in the scrambled situation. So what we did was to electroporate E35 ferret embryos with our constructs, and that let them survive for a couple of days to analyze what is happening to these cortical progenitors. And you can see here the result of this experiment. And the first thing we noticed is that the distribution of the electroporated GFP positive cells was very different, with a retention of cells in the ventricular zone and a loss of cells in the subventricular and intermediate zone. The next important thing that we noticed was the mislocalization of mitotic cells. As you can see here, again, where they're not located in the apical surface as in the control situation, but they're massively delaminated into subapical position and also basal positions, as you can see here. Now, this change in the localization of mitosis suggested a change in the polarity in the morphology of radial glia cells, which we could also somehow hint in these low power magnifications. When we looked in greater detail, indeed, the overexpression of this microRNA, the loss of function of this microRNA caused a very severe loss of the typical polarity of these radial glia cells, which lack their apical and basal processes anymore. And that was most likely the cause of this abnormal location of mitosis. Now, because also the loss of this microRNA in ferret cells will cause an increase in the expression levels of APC protein as predicted, because now we're sequestering the blocker of APC. So we thought that maybe by overexpressing APC directly, we might see even more enhanced the effects of losing this microRNA. So this is what we did. We, in this case, electroporated our ferret embryos to overexpress APC, and with the hope that we might find a similar phenotype as we have observed with the loss of function of the microRNA. And that's actually what happened. What we observed is that the location of GFP cells was again very much disturbed, and also the location of these mitotic cells. You can see here quantified a very massive accumulation of GFP cells in the ventricular zone and a very massive reduction of apical mitosis that were now localized in subapical positions. And actually, when we quantified this, we saw a very severe reduction in the total abundance of mitosis as a reduction in proliferation of these radial glia cells. And again, when we looked at the detailed morphology of these radial glia cells, as when we had performed the loss of function of the microRNA, now by overexpressing APC, we saw a very severe loss of this polarity of radial glia cells, which acquired a much more multipolar morphology. Now, finally, obviously, we were interested in the human situation, and by looking at published data sets, we observed that the levels of APC expression in the embryonic cortex are much higher in the mouse as compared to ferret, macaque, or human when we look in different layers. And when we look at particular cell types in single cell data sets, we also observed that the levels of APC expression in mouse cells are much higher than, for example, in human. Now, however, we still see some levels of expression of APC in human cortical progenitor cells. So we wanted to see what happens if we even reduce this further, if we increase even more the expression levels of the microRNA in the human cortex. So to do that, we turned to, obviously, human cerebral organoids, where we electroporated our constructs. And when we did this for our control construct, the scramble sequence, we could see this is an example organoid with a number of ventricles. And you can see here in red labeled the apical surface of these ventricles with PAR3, as before with mouse. And you can see that these ventricles have apical surface that we could measure. And this is the number of ventricles that we would observe on average per organoid, and the accumulated length of PAR3 apical surface on these ventricles. Now, when we overexpressed our microRNA in these organoids, we found first that the number of ventricles increased very significantly, almost twice as in the control organoids, and then also the surface area of these ventricles increased very dramatically, indicating that there had been a very significant amplification of apical radial glia cells that maintained their polarity in this case, and they highly amplified so that each ventricle was larger, and also we had a larger number of ventricles. So in summary, what we have in the normal situation is a number of radial glia cells that self-amplify and produce intermediate progenitor cells, and they're attached to the apical surface by the apical adherent junctions. With an expression of miR3607, or concomitantly with a loss of function of APC, what happens is we have an increased amplification of these apical radial glia cells with increased proliferation. And sometimes this is pathological and falls a little bit or generates an abnormal apical surface in these cortices. And this might be something that will lead to expansion of the cortical neuroepithelium. Now, in the loss of function of this microRNA, we have the opposite situation. We have first a loss of apical radial glia polarity, and we have also a loss of their proliferation. So this ventricular zone of this cortical neuroepithelium remains small. So what we think happens is that early in some mammalian ancestor, miR3607 was expressed in the developing cortex, and this was repressing APC. And so this repression of APC unrepresses or de-represses the activity of the beta-catenin pathway, leading to the proliferation of apical radial glia cells, increased expression of PAC6, and the delamination of these polar radial glia cells to delaminate and to establish basal radial glia cells. However, some of these species later on evolved to repress the expression of this microRNA in the developing cortex, like, for example, is the case of the mouse. Now, by repressing the expression of this microRNA in the developing cortex, APC expression was de-repressed, and so this came to the repression of beta-catenin signaling. So there was a loss of progenitor proliferation and delamination. And so we end up with a much smaller germinal layers with fewer and less amplificative progenitor cells, leading to the smaller and smooth cortex in the mouse. Okay, so that's it. Thank you very much for your attention. I just want to mention the main characters in this story. Mostly this has been the work of Kavya Chinapa, which again, she was a fantastic PhD student in the lab. And with some of the experiments, she had the help of Adrian Cárdenas, also Esther Llorens and Yuki Nomura at the technical side, Ana Villalba, with the electroporation of ferret embryos. She's now postdoc with Simon Hipp and Meyer in Austria, and also Rafael Soler with some of the dataset analysis. And again, thanks to our collaborators, Jose Lopez Atalaya and Angel Marquez, for their initial transcriptomic analysis. And obviously, thank you for the funding bodies. And that's it. Thank you very much.